Good afternoon, everyone. So happy to be here with you. My name is Bettina Hamelin. I am the President and CEO of Ontario Genomics, and I welcome you to our panel on a Canadian engineering biology roadmap for the building of a biomanufacturing future for Canada. And I want to apologize for the slight delay. We had some technical issues and we'll have to manipulate our slide sharing ourselves. So please bear with us, but we're really thrilled to be here. An amazing panel who will share some really exciting and urgent opportunities for Canada, while also having a candid conversation about, our challenges and our risks that we have to consider when innovations are as disruptive as engineering biology can be. I want to just briefly thank the Canadian Science and Policy Conference for your ongoing work to bring government, academia, industry, so many stakeholders together for some really important conversations. It's already been such a great conference, and I hope you didn't miss Genome Canada's pre-conference panel on the future of food, very much aligned with the discussions we're having today. And also want to point you to a session by Genome BC on November 18th on their collaborative and cooperative response to COVID-19. So lots of exciting stuff happening. With regards to our panel, we really have two goals here today. First of all, we want to share with you why we are so excited about engineering biology and its potential for a green economic recovery, where we don't content ourselves by just merely rebuilding our losses, but we're working towards a new economy, a new economic future for Canada as part of a global biorevolution. Second, we feel strongly that Canada needs to recognize that engineering biology is one of the disruptive technologies of the 21st century. And it requires focused support from government in collaboration with industry and all the stakeholders in this ecosystem to make it happen together. And so we hope that we convince you today that this is a very important cause and you will join us and support us. So I will share my slides with you, and hopefully that works. Hopefully you are able to see my slides now. Really, I want to give a bit of an introduction. We know that some of you might be new to the topic. And by the way, we wanted to do a poll, and apparently the technology cannot support that today. But we assume that some of you are new to this, some of you know a little bit about it, and some of you are like our panel members who have dedicated their careers to it. So we're really looking forward to engaging with all of you. But I wanted to start by introducing engineering biology a little bit. And please do use the Q&A feature. Send us questions. We will address the questions during the second half or third of the session today, so that we can hear from you and hear what questions you might have. So over the past 20 years, we have made incredible progress in our understanding of biology, biological sciences, genomics, and molecular biology. And that's why today we can not only sequence and understand the genetic code, or the genetic code of a virus, for that matter, we can reproduce it, we can write it, we can edit it. And that's been taking us into a completely new era, an era where engineering biology can really be used to make useful stuff. And by the way, we're not really inventing things here. We are merely discovering the biology that nature has brilliantly invented millions and billions of years ago. And we are finally catching up, understanding how this works. And that helps us reproduce parts of it to reinvent how we make the things that we want and need. But humans have used biomanufacturing since the beginning of civilization. You see here, the natural bioprocesses was used by employing yeast to make wine and beer for thousands of years. Now, thousands of years later, we're still pretty much using the same process to make biofuels. We have been able to industrialize that process. But where it gets really exciting is that now we can do things at scale. And we can do that because there's this convergence of these natural processes with our ability to program cells as factories, use AI-driven design, and high throughput automation that, even 2 decades was not possible. And all of this has allowed us also to reduce the cost, and that's really changing the game and drives biomanufacturing into this revolution to make things like medicines, plastics, clothing, food, all the things that underpin our society. Now, here are just some examples of products that are possible with engineering biology. And I think we're all observers of how fast the field of vaccines has moved based on COVID-19, where scientists were able to sequence the virus within weeks rather than months. And that has allowed pretty much immediate development of diagnostic testing, which is based on the viral genome, and has also allowed the design of vaccines based on the virus. And these vaccines have entered clinical trials at rapid speed that we have never seen before. But whether it's a novel vaccine that's entirely designed based on a viral genome, or whether it is a plant burger that contains heme, which is made by yeast that's been engineered to express the soya heme gene, or whether it is alpine genes that are made by fermenting microalgae to produce a completely new oil, an oil that cannot be made by synthetic chemistry, but a completely novel oil, and that can be turned into polyurethane and combined with aspen to make high-performance skis. So some really cool stuff is happening, and I could fill all matters of slides with this incredible products. And what makes that really possible is that people are interested, people want and demand these products because they also correspond to their values. And investors have been seeing this, and on the lower part of the slide, you see global venture capital and private sector investment into biological technologies in 2018 amounted to $20 billion and far exceeded investments into other disruptive technologies that we all think are probably pretty cool. So earlier this year, the McKinsey Global Institute published a terrific and well-researched report that I recommend to everybody. And at the crux of the report is really the prediction of a very much forthcoming biorevolution. And that biorevolution is rooted in the fact that as much as 60% of the world's physical inputs could be made by biological means, replacing oil and gas and petroleum, but using biology to make these inputs. And that could lead to $2 to $4 trillion in economic impact by 2030, 2040. So we're not quite there. We're definitely headed there. And that's really completely transforming our economies and our lives, including what we eat and what we wear, the medicines we take, the fuels we use, and quite frankly, how we construct the world in which we live. But we need to do this right, and new innovation, disruptive innovation, and these new biological capabilities really come with profound and unique risks that we need to manage proactively. And today we'll be discussing this for sure. Now, other jurisdictions have recognized the power of engineering biology decades ago, in particular in the US and the UK, but Australia, China, Singapore, Japan are really on this, have been on this path for quite some time. And what we're seeing now is that these engineering biology consortia that develop these roadmaps are now at the stage where they want to scale up technologies and are creating, very state-of-the-art biomanufacturing and biodesign operations, like the recently announced Biomade in the US. And we can see in the UK that has been investing in synthetic biology, which is another term for engineering biology. And we can see how they're really benefiting on a commercial scale in terms of deals that they're making. Now, the great news here is that we don't have to recreate the wheel. A path has been set. We don't have to make the same mistakes others had to make, just because it is hard to develop these new innovations. So we can learn from others, and we can fast forward to really create our Canadian solution. And we would say that there's urgency and that the consequences of not acting are dire. And it's really highlighted in this McKinsey report what the opportunities are. There are more than 400 of them. And so every sector of our economy really is affected. And we need to get ahead of that. And not only do we need to support, get the pipeline going, support our startup companies, but we also need to equip our traditional companies with the kinds of innovative approaches and transformative biotechnology approaches so that they can compete globally. And what I'm really excited about is that the government has really recognized the importance of biomanufacturing and has made critical investments. And you'll hear about some of this today. And now the opportunity we have is we must leverage this for the broader economic impact. But we are really on the right track here. And what we need to make sure is that we really build on this momentum. So we've been very active in bringing the engineering biology community together over the past few years through our Canada SynBio conferences and workshops, and that has sort of evolved in the creation of a National Engineering Biology Steering Committee. And I think you'll agree with me is that there's a really great expertise and experience on the steering committee, so that we're in really good hands as we are formulating our strategy going forward. And you will hear from some of our steering committee members on a panel today. And essentially, this committee has focused on really looking at what are the opportunities, what are the specific opportunities in Canada in this field, and has really focused on two big initiatives. The first one was the establishment of a Canadian engineering biology ecosystem or network. We called it Can Design, together with more than 85 partners from industry, from across sectors, from across disciplines, from across the country. Really the Canadian response to this movement that we've been seeing around the world. And of course, we're building on the great advantage that we have all the ingredients, whether it's the world leading academics, whether it is innovative companies, whether it is the biofoundry infrastructure that is emerging. We got all the ingredients. What we need to do is organize ourselves so that we don't fall into this, our traditional fragmented approach that sort of hinders the realization of the big potential for Canada here. And so this cohesive network really connects the right players with the right people to really think about big problems, and the kinds of big problems that benefit not only from the ability to bring the players around the technology together, but the kinds of problems that really benefit from cross-pollination, that benefit from experts. For example, experts in the production of advanced cell therapies can accelerate technology development for those in cellular agriculture. So sometimes the inspiration for innovation doesn't come from your own sector, but it comes through cross-pollination with other sectors. And so Can Design was really conceptualized to do exactly that. And the second piece is a white paper on engineering biology, and we're thrilled that coinciding with the conference today, we're launching this white paper that was co-written written by the National Steering Committee. It really highlights the opportunity. It highlights what are those 3 focus areas we should be taking leadership on, and we can come back to that. But the paper is also first and foremost a call to action. And the call to action is really for us to come together and to ensure that there is funding for an inclusive engineering biology innovation ecosystem in partnership with industry that leverage all of our current and future capacity across industry, academia, and government, so that we can accelerate products that we will then export to the world. We need to be building on recent investments in vaccine development and manufacturing to include, these multifunctional biofoundries and scale-up facilities. We need to create the right legal, ethical, and regulatory conditions. We need to support outreach and community engagement, so we gain the trust of the consumer, and these novel technologies can get adopted. Obviously, major implication for jobs, so we need to develop the talent and skills. And last but not least, we really need to seize the opportunity for collaboration and partnership across funders, across organizations in Canada, nationally and internationally. And with that, I'm thrilled to introduce our super panel who's joining me today. And we will hear first from Dr. Peter Zandstra, who is the director of the School of Biomedical Engineering, Michael Smith's Laboratories at the University of British Columbia. We will then hear from Lakshmi Krishnan, who is the acting vice president, life sciences at the NRC, followed by Vincent Martin, who runs a biofoundry and is prof at Concordia University. We'll get a private sector perspective from Adam Westbrook, who is metabolic engineering lead at Genecis Bioindustries. And then Vardit will round it up on the so important ethical implications of all of this. And Vardit is a professor at the University of Montreal. So with this, your slides. Peter?
Yep. Hi, everybody. Can you see my slides? Excellent. It's really fantastic to be here. Thank you very much, Bettina, for that introduction. You really set the stage well for I think the discussion that's to follow. I'd like to focus my comments today, my introductory comments, on a specific sector of engineering biology, cell and gene therapy, as really an example of Canadian leadership and where we can take the foundational science and the translational expertise that we have and really make an impact. So I'm just going to go forward to my next slide. Great. So one of the things that's important to recognize is we're not talking about some future many years away. The products of engineering biology are really very much being applied today. So if you think about the vaccine, for example, from Pfizer and others, that's really an example of a technology where it's a drug combined with a lipid nanoparticle. It's engineered to have certain properties and is being delivered. And as we think about different drugs, biologics, and now cells, we're really moving up the spectrum from natural product to synthetic drug and eventually to an engineered combination product. And this is true with simple drugs as it is with biologics, where we're now moving to antibodies with bifunctionality that just don't exist in nature but are designed around specific clinical need. Similarly, as I'll highlight today, we're doing the same thing with living cell therapies, this fourth pillar of medicine, where now engineered cells are starting to act in specific ways according to the design principles which we're putting them. So just quickly, I think Bettina already hit on this. One of the things to recognize is this is also a great economic opportunity. There are thousands of clinical trials underway, including very late stage clinical trials for cell and gene therapies. And in 2020 alone, so far, there's already been a significant increase in investment in the area, with over $10.7 billion in venture funds put into cell and gene therapies this year. So that really tells us that not only internationally, but also in Canada, we're starting to see great opportunities around the early stage products that are emerging out of this. And this is an example from the Canadian cell and gene therapy landscape, where companies such as those listed on the side have already achieved investments and are moving new products forward based on the cell and gene therapy landscape. One example that I'd like to highlight today quickly is the example of cellular immunotherapy. So cellular immunotherapies are engineered cells that are modified to express cell surface receptors which target certain types of cancers. These so-called CAR T cell therapies have been shown to have really good efficacy in some types of cancers, and really raise the question as to how we're going to ensure that Canadians have access to these types of technologies, right? Especially as they start expanding the different types of indications. And one of the, there's a number of limitations with these technologies currently, as we can see on this slide, but one of the biggest ones is the cost per patient, with current therapies costing almost $1 million per patient. Now you can imagine for our healthcare system, we need to find solutions to this type of problem. And those types of solutions can really be found in the innovation that is around engineering biology. So just to give you a sense of what we've done in the past and where we are now, I think significant investment has been put into understanding the structure, function, and the parts of cells, and how those cells and the components and networks in those cells are related to each other. I'll call this reverse engineering, where we're trying to understand how biology works. Where engineering biology has an opportunity, and where I think we need to understand more in the future, is this forward engineering aspect, where we now take these parts and these components and start to think about how we can manipulate them in a strategic way to get the outcomes we want in terms of cells and tissues. So when we think about a cell, there are many handles we can have to start to manipulate these cells. These handles can include the sensory inputs, whether they be signals we provide to the cells, the cell surface receptors that we put on them, or the disease markers associated with them. Inside cells, there's a logic processing circuitry, which we have more and more control over putting orthogonal pieces into. And finally, as these cells start to have regulatory functions and outputs, we can make these and connect them to different drug aspects, whether it be secretion of molecules, changes in protein activity, or changes in gene expression, which might be associated with different genetic diseases, for example. This really allows us to start moving forward to a whole new paradigm in medicine, which is instead of starting with what we are able to produce, we can turn the question around and say, given unmet clinical needs, how can we start to design solutions based on manufacturability, based on the quality of the product, and based on the potency that will allow us to drive forward towards affordable new biologic therapies. And that's indeed what we're starting to do in this area of immunotherapies. We're now, for example, by producing these cells from stem cells, using scalable systems, using gene editing, which is done at different stages of development, we can imagine a future where we're significantly able to reduce the cost of these therapeutics and increase their accessibility. So I just wanted to provide a few examples of that today. I look forward to our discussion. In the health area, I think engineering biology gives us a unique opportunity to connect our health priorities to health impacts by starting to reduce the barriers between the traditional health areas of devices, therapeutics, and diagnostics, and really start designing a priori molecular and cellular systems to solve these problems. So with that, I'll pass it back to Bettina.
Thank you. Thank you very much, Peter. That was such a great overview of the opportunity here. And I wonder, Peter, whether you could just unpack a little bit as we're transitioning to Lakshmi, the potential benefit of these types of cell therapies, when we compare it to chemotherapeutics that are, still largely used to treat patients and are associated with all these adverse events, etc. What's is the promise so that this is, more targeted and more tolerable as a therapy?
I think we're still addressing some of the elements of the therapy in terms of their safety relative to chemotherapy and other things. But one of the aspects that's really important to think about is these are not therapies that are that are really starting to target towards cures. And what I mean by that is cell therapies are living cell products that are able to address more of the long-term fundamental changes which are required to prevent the disease from remanifesting. So in immunotherapies, that's really about active T cells with memory capacity that are able to regulate and keep the cancer from reemerging. In diabetes, for example, as another one, we're talking about replacing insulin cells, insulin secreting cells, as opposed to masking the effects of the lack of insulin in that place. So I think we still have a number of challenges to move forward in terms of the overall best design of these systems, but the early indications are from a variety of different therapeutics is that we're going to be able to overcome these problems.
That's really excellent. That outlook for the longer term and the cure would make a world of a difference for sure. So thanks, Peter. And Lakshmi, I see that you're preparing your slides to share.
Yeah. Do you see my slides now?
Yes.
Perfect. Okay. No, that was, thank you, Peter and Bettina. Those were great introductions to what I wanted to kind of touch upon from the National Research Council perspective, where we are investing in engineering biology. So just for those of you who may not be absolutely familiar, we are actually 102 years old. And I think there are two arms to what we do. We are a major research performer in Canada with 14 research centers, which constitutes our intramural research, as well as we have the program which funds small and medium-sized industry through the Industrial Research Assistantship Program. And I think this is a kind of a metric slide, but what I wanted to make a point here is that because we have research centers in many different sectors, it lends itself very nicely to collaborative programs. And in the area of, for example, engineering biology, this will be very evident for you as I talk through my presentation today. So in terms of our, foresight, I mean, what we looked into, what we can expect in the years to come, particularly given the current pandemic and what has happened and what has transformed in the wake of COVID, it's very clear that COVID and health innovation are going to be something we absolutely will need to prioritize. And responding to COVID will not only become a national priority, but it also is going to have an impact on health innovation in a number of associated comorbidities, particularly rare and chronic diseases. And in our mind, I mean, these are areas which, again, very well lend themselves to innovation in engineering biology to not only be able to bring innovative solutions, but also to increase accessibility for these in the larger Canadian population. A second area we think there's going to be quite a bit of activity is food security, particularly in terms of food production innovation and also moving towards automation and biotransformation. I mean, these are again aspects that we think not only will sustain agriculture, but also is an area where there will be active engineering biology solutions that are pioneered. Climate adaptation is a third pillar that NRC is looking into prioritizing, and all the different impacts of the global warming, and again, innovative technologies in this space. And in some of these environmentally friendly solutions, you can again see some of the engineering biology solutions that will certainly make an impact and also will have significant impact in the future. And lastly, underpinning all of these first three areas will be the ongoing digitalization of both society and economy. And here again, using aspects such as AI and machine learning, quantum, and adoption of new technologies, we can bring forth these solutions in engineering biology. And you will see a flavor of that in many of the presentations you will hear today, I'm sure. So from our side, the way we have approached this is, first of all, we have really approached it from collaboration lens. And so one of the big programs that we are right now investing in is the disruptive technology solutions for cell and gene therapy. And within the scope of this program, we're really looking to bring forward disruptive technologies to enable affordable engineered cell and gene therapies. And this, we want to do it in collaboration with the various sectors in Canada. So our aim then is to not only accelerate the discovery cycle for these cell and gene therapies, but also help establish the critical ecosystem and capacity for distributed clinical product production and engineering and GMP manufacturing. And in fact, we have done some investments in this front where we are enabling groups to be able to invest and grow in this area. Including the gene foundries. Of course, optimization of the biomanufacturing process is going to be extremely important, particularly if you want to have cost-effective solutions. And then ultimately unifying the Canadian ecosystem for collaborative delivery. And I think this is an area where Canada has a very strong discovery pipeline as well as a very good ecosystem. So I think the challenge in front of all of us is how do we ensure that those benefits are accrued in Canada with a Canadian footprint. So some of the aspects in this program we're looking at is innovative platforms. So one example, for example, is using high throughput pluripotent stem cells or iPSCs for gene editing. We're also looking quite a bit on using technologies such as microfluidic devices for increased automation, and also, of course, AI for design. And I'll talk a little bit about those aspects in the coming slides. This program also has been really tending to support the Canadian ecosystem. So we have done infrastructure support through grants and contributions. So for example, some of that is listed here in the first tranche that we have supported to expand the capacity in Canada. And we hope to increase this type of activity further along in the 7-year program as we progress. The strengths really of running this type of a challenge program is that we can bring intramural and extramural research together. And as Canada, we can have a significant impact on bioprocess engineering and product development and characterization. And in fact, so I will end by saying that there is certainly a lot of opportunity within this public-public or public-private partnership models for rare diseases. And this may be a new lens we have to apply in order to make sure that the impacts of Canadian discoveries are fully realized in Canada. So I think Peter talked a little bit about it, and this again reiterates that one area, for example, is the CAR or cell therapy. And I think the opportunity here is quite ripe because on the right-hand panel, for example, you can see that there are many formats you can actually assemble using these multi-modular antibodies. And so at the NRC, for example, we have an armament of antibodies that we're developing, and then you can actually assemble it in various ways to achieve precision targeting or even multi-targeted CAR. And ultimately even a model of off-the-shelf therapies where it is not person or patient specific, but it can be more broadly applied in types of a universal cell type. Of course, one of the projects, for example, right now that we are advancing quite a bit is in collaboration with BioCanRx and other collaborators in BC, where we're looking at a single domain CD22 made in Canada CAR. So single domain antibodies, for those of you who are unfamiliar, are really very small modular antibodies. They are actually only made by camelid species in nature, but we can certainly not only immunize camelid species and pan antibodies against relevant receptors, but you can also, use a human library to obtain these very small sized antibodies. And these lend very well to these multi-modular formats, and using the engineering biology approaches, we can definitely increase the precision and targeting of such new therapies. A second area where we are also looking quite actively, and we feel would be a very important area, is to demonstrate the capacity of microfluidic based device technologies to enhance the development of next generation cell and gene therapies. And essentially the idea is to automate the production for those micro doses, and also both this could be used in the context of either autologous or allogenic therapies. And the power here is that by using this type of microfluidic approach, as opposed to the current approach, which is shown here in centralized manufacturing, where you have to get the patient cells, you send it away somewhere, you have to now reassemble the engineered cell and back to the patients. There's quite a bit of steps and logistics, and also the cost really comes from that approach of trying to do this in that type of a model. Whereas our vision is that by using a decentralized microfactory right at the bedside, we might be able to achieve that in a more efficient and a more precise way. And in fact, one of the technologies that was developed at the NRC is a power blade technology, along with some microfluidic prototypes that we have developed. We are now applying this to manipulating cells at the bedside and understanding the key criteria and trying to use this approach in the next iteration. And so the aim then would be to have decentralized GMP grade operation, which can be customizable. And this miniature hardware, of course, can be AI monitored, which would give a one extra step of benefit. And we can also apply it for online cell analytics as cells are being grown in reactors or at the bedside, in order to ensure a precision on the type of cells that are being transferred to the patient. And this type of technology would certainly be low cost reagents and much more affordable than the current technologies at play. The last area I want to talk to you about is agri-food innovation, and I apologize for the typo there. It's not food, it's food. And the application of systems engineering biology to agri-food research. And the first aspect here is modulating or introducing metabolic or signaling pathways in crops. And this is an active area of research for us, particularly in Saskatoon with our protein supercluster, as well as with the University of Saskatoon, where we are trying to optimize traits impacted by climate change or improve sustainability. So this can then impact either primary production or resource efficiency, as well as resistance to emerging biotic stresses. And one of the other aspects here is that we could improve seed composition by applying engineering biology process and optimize the key development stages for this type of nutritional approach. The second area is introgression of multiple signaling pathways from wild relatives into adapted crops to optimize nutrition profiles. In fact, the canola itself is actually an engineering biology product that was pioneered by NRC many, many years ago. And we know today that that is one of the most economic drivers, as well as nutritional value. And introducing and adjusting metabolic networks to microbes is another area that is emerging and is of great interest to us, particularly to permit the biotransformation of agricultural co-products to more value or added products. And also to improve the protein quality and functionality, which again is something I think with an engineering biology approach we can achieve very effectively. The last slide here, I want to just emphasize that all of this will not be possible without building biomanufacturing capacity. And as Bettina alluded to in her presentation, this is a very important area of investment that Canada is making and is continuing to make. And one such project that is currently ongoing at the NRC is our expansion of biomanufacturing facility in Montreal. And the way we look at it is biomanufacturing is a continuum from early clinical trial material production all the way to large-scale production, formulation, fill finish, packaging, and logistics. So depending upon the product, we really have to keep the entire life cycle in mind and customize it as it would be appropriate, either for a point of care biomanufacturing or, for example, biomanufacturing biologics and vaccines. And so we have embarked on this, and we are actually putting together two facilities. One is a clinical trial material facility, which would have the capacity, particularly for viral vector production at 500 liters, and also a larger biomanufacturing center that we hope will have the capacity of 4,000 liters of GMP biomanufacturing. But I'd like to emphasize, I think this is still just the first in the series of investments Canada is going to need to really bring forth biosecurity in this space. So with that, I think I will stop my thoughts here on how engineering biology can help sustain the bioeconomy in Canada. Thank you.
Thank you so much. A great illustration of how, engineering biology is this platform that serves, multiple application, and you showed health application and ag applications. And I think I'm going to move to the next presenter in interest of time, but I think a question that people are interested in is how do we work with the NRC to be able to leverage all this technology that's being developed. So I'm sure we're going to come back to that. A note to the audience, we've been informed that the Q&A feature does not work in the platform today. So I encourage you, if you have questions, please do send them via email to myself at bhamelin@ontariogenomics.ca. bhamelin@ontariogenomics.ca, and we'll see whether that works. In the meantime, let's move on to Vincent with his presentation, and I will share my screen. Let me see. Vincent, does that work? Do you see the slides?
I see myself, but your slides, I think, are in the backstage. Here we go. Now I see your slides.
You see them?
Good.
Okay.
Yes.
Just let me know to move them.
Yes, sure. You can skip to the next one. We've been introduced properly already. So I think that we had a very good introduction from the three previous speakers on engineering biology and the benefits that engineering biology can bring. The point I want to drive home more today is the fact that if we're going to use biology as a technology platform, we have to think about ways to make it easier and faster to engineer to get to the products or the strains of the cells that we're trying to do. And in order to do that, you really require three things that are central to mass uptake of innovation. One of them is standardization. The example I give here is nuts and bolts for the car industry, let's say, or CPUs and different chips for the computer industry. You can actually use a bolt on different cars, and you can use the same chip on different computers. So that's the standardization aspect. The abstraction is very important. And coming back to standardization, we all use a Lakshmi promoter to express things in E. coli. And the reason we can do this is it's really well characterized and it's standardized. The next important point in moving innovation to mass uptake is really abstraction. And this is an important point. And it's illustrated in this figure here in the automotive industry, but the same holds true for biology, where you don't need to understand how a cell fully functions in order to use that cell. We do that every day. Or you don't need to fully know how an enzyme or a protein functions in order to use that protein or that enzyme. So as long as you have really well characterized parts and devices that you can use, you don't have to fully understand how they work to use them, either yourself or the end user of your device. And the last part is really the automation. If you're going to be able to scale a lot of these things, whether it's through iterative designs and testing or through scaling up, you really need automation to come to the picture. So when we think about picking up biology as a next innovation vector, we need to think about things like standardization, abstraction, and automation. So if we go to the next slide, I have examples that should be obvious to everyone here. But until we have standardization and automation in the car industry, through the oil, gas, and steel boom that we had, we were building cars one by one, and they were sharing the road with the horses. And it's only through standardization, abstraction, and automation that we were able to mass produce cars, and ended up with what you see on the bottom right, a mass uptake of that innovation. On the next slide, the same is true for the silicon and semiconductor industry and the obvious impact that it has today. Until you had standardization, abstraction, and automation, you really were not able to get mass uptake by the general population, and we did that, and now everyone carries a small computer in their pocket. So if we go to the next slide. So the question I always have is, how to use biology as the next technology vector? And I put two pictures on the right intentionally there. One of them is Pasteur in the 1980s, and the other one is today's scientists, and they look very much the same. And even though I know the oil and gas and steel and the silicon semiconductor industries have managed to advance their technologies and mass produce these things through various innovations, we still view biology very much as a very labor-intensive proposition. And a lot of us still think about tinkering and discovering. As much as we'd like to think we know biology really well, there's still a lot that we don't know. And a lot of us still consider biology an art more than a science. And this is really what we should be thinking about when we think about using biology as a vector for innovation and mass uptake. So if you go to the next slide. Oops. So the question that I keep bringing up all the time is, how do you industrialize something like biology? Well, the simple answer is you really have to start applying the same principles as engineering. And unless you do that, you're always going to be stuck in the tinkering and playing around with different pieces of DNA and different cells, and it's always going to be difficult for you to shorten your time of development and really scale your discoveries and your innovation. So if you go to the next slide. So this is an example of our ecosystem at Concordia that we developed to try and address these questions or these issues in industrializing and scaling biological engineering. We have really a 4 or 5 prong approach to it. The first one is we have a research center that brings researchers from across Canada, not just Concordia, who are interested in biological engineering, and we share ideas and technology and innovations. We have a technology platform, which I'll talk about in a minute, which is the Genome Foundry. We have now a startup accelerator program to allow these lab discoveries and young scientists and entrepreneurs, budding entrepreneurs, to actually take these ideas to the market. We actually have a training program that's funded by the NSERC Create program for budding biological engineers. So we teach exclusively synthetic biology and biological engineering kind of courses and program. And finally, we're developing partnership. One of the big ones that we've just announced recently is a partnership with NRC. You heard a little bit about it in the previous talk, where we're developing a technology platform, or in the Genome Foundry, a process for high throughput iPSC cell engineering. Next slide. So what do I mean about a Genome Foundry? And this is a concept I want to describe a little bit. What does it do and what is it for? So really what a Genome Foundry is a fully automated end-to-end solution from the design to the construction, testing, and analysis of a user-defined genome. And really the goal is to make biology easier and faster, more predictable and scalable biological engineering. And we do, and this is really the important part I think that I'm trying to drive home in my presentation, is until we get to the point where we can have easy, fast, predictable, and scalable designs, it's always going to be tinkering, and it's going to be hard to go from the bench to the market in a fairly short time span. The design, build, test, learn cycle, Bettina introduced already. This is central to a genome foundry. We have this at Concordia. We have all of the different steps from designing whatever device you want, whether it's a protein or a cell or a gene circuit, to building that circuit, that cell or that protein, to testing it, so phenotyping your device. And we're slowly introducing machine learning and artificial intelligence in the process. And that's really key to closing the loop and accelerating your design, build, test, learn cycle. Next slide. So even though it seems to be quite a out there idea to build genome foundries, we're not the only one. So far, there's at least 6 full-scale genome foundries, or 16, sorry, not 6, 16 genome foundries that have been built across the world. And we actually created a Biofoundries Alliance a couple of years ago. And the purpose of this was really to exchange information on automation processes, on standardization, so that we can exchange parts, devices, cells, genes, and proteins across the foundries and have one unified system to go on. So this is really central to what we're trying to do, and it's really valuable to our foundry and the foundries across the world. There's actually examples here on this map of for-profit foundries. So I think there's Ginkgo's on there, and the Agile Biofoundry and Zymogen on the West Coast. But a lot of these foundries are actually state-run and state-supported, and their main purpose is really to develop innovation and bring things to the market as fast as possible. So we have competition across the globe in this sector. Next page, please, or next slide. So in closing, I just wanted to give a couple of examples of partnership that we have in our center, and we're actually looking at different verticals. So we're looking at food and beverage. So that, and when I say food and beverage, that includes ingredients, not just food that you directly consumed. We have activities in the chemicals and fuels sector with BP and L'Allemand and FP Innovation. As I mentioned already, we have our partnership with NRC, and we have a couple of partnerships with drug manufacturing company, whether it's in the analgesic space, and more recently with a company interested in cannabinoids. So we're really using our platform to bring activities and help companies, whether it's private, public, or academic, to really speed up their process of biological engineering and get things to market as quick as possible. And next slide, please. And in that, I just want to close, and you have my email address there. So if anyone has any questions or want to have a follow-up discussion on what we do in the foundry and at Concordia, please don't hesitate to reach out to me directly. Thank you.
Awesome. Thank you very much, Vincent. That was great. And, you know, it sort of reflects some of the things we've been talking about in terms of, you know, how engineering biology is a platform for multiple verticals that you're working with. And partnership is really crucial. It's partnerships with the industries, it's partnerships with the accelerators and startups organization, and partnership internationally. So that was really a great summary of where we are at there. Next is Adam. And Adam, I think you can share your slides directly. Wonderful. Over to you.
Okay, can everyone hear me? Good afternoon, I'm Adam Westbrook. I'm the Metabolic Engineering Lead at Genecis Bioindustries. And we're a relatively new company that's focused on upcycling organic waste into affordable premium chemicals using proprietary bacterial cultures. And so I'm going to talk briefly today about how we're using engineering biology as a company to solve a problem that's obviously of great interest to us, but also to the entire world. So we likely all have a kind of a love-hate relationship with plastic, right? It's cheap, it's very durable and inert, but it's basically forever. And the promises that plastic producers have made about recycling plastics are not being realized. I mean, in the US, basically about maybe 8% of the plastics that are produced per year are being recycled, whereas about 3 to 4 times that amount is actually going to landfill. So it's not happening. Another great example of how plastics are really negatively affecting our planet is the Great Pacific Garbage Patch, which is literally a small continent of plastic in the Pacific between Hawaii and California. It's now twice the size of Texas, and it's growing every day. So in response to this glut of plastic, companies, a lot of major corporations are trying to replace petroleum-based plastics with bioplastics. In particular, there is one called polyhydroxyalkanoates, or PHAs. They've been around for a very long time. We've known about them for a few decades. And they're considered kind of the platinum of the plastic industry because they're biodegradable. You can tailor the properties so you can use them in any number of applications, and oftentimes have better properties than petroleum-based plastics. But of course, they are expensive. And that's the issue we're trying to resolve at Genecis. So traditionally, PHA has been made from sugar and oil feedstocks that are derived from agricultural feedstocks like sugarcane, canola oil, palm oil, corn. And of course, this is a dual issue because, first of all, it's not sustainable. We're competing with food crops, and we also need a lot of arable land to meet our insatiable plastic demand. So this is just not going to work long term. So at Genecis, we are trying to address this issue using a sustainable approach. So we've developed a technology to produce PHAs from organic waste using mixed cultures, mixed microbial cultures, which can reduce production costs by at least 40% to hopefully make these plastics, these bioplastics, kind of more to be able to compete with petroleum-based plastics. So we can hopefully replace them at some point. So our current process is comprised of 2 fermentation stages. Okay, and we've been working on these mixed trade secret bacterial cultures for over 3 years to try and improve the performance. We use volatile fatty acids, or VFAs, as the carbon source for PHA production. They're produced through anaerobic fermentation of food waste in stage 1 of our process. The VFAs are then fed to another type of mixed bacterial culture, which can produce PHAs intracellularly. We can then harvest the PHAs, and they can be used by companies to make products. Now, we're currently focused on a particular copolymer, which is called poly-3-hydroxybutyrate co-3-hydroxyvalerate, or PHBV. And it's made of 2 monomers. That's 3-hydroxybutyrate and 3-hydroxyvalerate, or HV. Now, the composition of our PHBV is tunable, so we can change the HV content by adjusting fermentation parameters. Now, by tailoring the HV content of PHBV, we can alter its properties for use in a variety of applications. For example, less than 5 mol% HV. You can make filaments for 3D printing, mechanical pencils, golf tees. More than 20 mol%, you can produce thin films for packaging, grocery bags, all this kind of stuff. So it's quite versatile. Now, getting on to where engineering biology comes into our process. In parallel with producing PHBV in mixed microbial cultures, which are kind of conditioned wild-type cultures, Genecis, primarily through my work, is developing engineered bacterial strains that can produce PHBV with a goal of obtaining tighter control over the properties and trying to improve the yield of the polymer compared to our mixed cultures. And so this is an excellent example of how engineering biology can be used to maximize the returns of a process that should be an integral part of Canada's circular bioeconomy. And not just Canada, the entire globe. This is an issue that's facing all of us. And in addition to producing PHBV, we're also engineering bacteria to produce other more valuable PHAs, such as those that are used extensively in the biomedical industry. And I'm also drawing on my experience in engineering VFA metabolism to construct other strains of bacteria using, again, engineering biology, so that we can produce things like antibiotics, vitamins, other specialty chemicals using catalysis. And again, all from low-value waste that costs nothing to us using an engineering biology approach. Okay, so now beyond the application of engineering biology to design bacterial metabolism for the conversion of waste to PHAs or other products, we can also use engineering biology to program cell physiology for simplified recovery of PHAs. One example is we can engineer bacteria to survive under very harsh conditions, and that way we can actually run our process under non-sterile conditions, which means we don't need steam and clean in place, which can significantly reduce our production costs. Similarly, we can also engineer cells to aggregate with each other after the PHA production process occurs, and then we don't have to use centrifugation to collect the cells, which is another big cost savings. And another approach that we're working on is to engineer cells that will lyse automatically once the PHA is produced, and thereby releasing the intracellular PHA so we can easily recover it from the medium, and then we don't have to collect the cells, we don't have to break them open, and we don't have to do the same extraction steps, which could significantly reduce our production costs. So I mean, we have many examples here of how, just to tackle one problem, there's so many ways in which engineering biology would actually either significantly improve the performance of a process, allow us to tune the composition of our product very finely so we can target a much wider range of applications for various companies that we would be partnering with, and also just ways in general that we could reduce bioprocessing costs, which you simply just could not do with natural bacteria. And another point being is that while PHAs can be produced in your sort of kind of a wild type culture, although they have been adapted quite a bit, but they're still kind of similar to a wild type culture. It's very unoften that you will be able to produce anything of interest in significant enough quantities using a natural bacterial cell. You'll almost always have to engineer the cell to either put the pathways in that are necessary to actually go from product, or from substrate to product, and actually, you know, even if it's a natural product, you will need to use cell engineering to get to the titers that are actually feasible for scale up and commercialization. But of course, you know, the way to go about doing that is evolving, as Dr. Martin pointed out. So it's not only important to understand that it's necessary, but that without developing these technologies further and automating them, then we won't be able to fully exploit the capabilities of engineering biology. I'd just like to point out, with regards to our technology, it has been validated by some reputable organizations like BASF. We've won some innovation competitions with them, recently also with Novo Nordisk, and we're also partnering with to develop some technology for them. We also received a big grant from the Sustainable Development Technology Canada. That's a government agency that's working to develop sustainable technologies. And we've won all kinds of other awards as well, as you can see at the bottom. And that's all that I have for now. Thank you.
Very cool. Thank you so much, Adam. And I guess as we're transitioning to Vardit, a question I have in light of the fact that you have to essentially, you know, engineer your microbes or your cells and in a customized way, does that open the door to different inputs, i.e. you're starting now with waste, but could you start, for example, with other types of inputs? I'm thinking, you know, 99% of the clothing we wear ends up on landfill. Could you use that kind of input to make PHAs?
Well, the clothes would be a difficult one. I mean, you know, certainly there's been a lot of work to generate carbon sources that can be used for fermentation from biomass. And there's been a lot of success stories there, but, you know, compared to a synthetic fabric, or probably even something like, you know, cotton, which is still kind of natural, they're fairly, I guess, unrefined. So to use something like clothing, I'm not saying it's impossible, but certainly, you know, that's not necessarily a simple problem to tackle. But I think, you know, with the automation of synthetic biology and engineering biology worldwide, and I just more funding being devoted to the field. I mean, anything's possible, but certainly that's a fairly big nut to crack.
Yeah. Okay. Awesome. Well, we need big nuts to crack always, right? To stimulate the innovation. Awesome. With that, I'm handing it over to Vardit to round up our panel presentations here. And we see your slides.
Great. You see my slides, that's wonderful. We don't have a lot of time left because we started late, so I'm going to keep my comments very high level, and hopefully we can still have time for some discussion. There are so many different ways to approach the ethical, societal, and governance challenges raised by the technologies that were discussed. So I want to start off by pointing out that my slides are not advancing, so, oh, good. By pointing out that too often when we think about ethics or bioethics in the context of emerging technologies, we frame our thinking in terms of why not? Why shouldn't we do this? We focus on the issues, the risks, the barriers. And I'd like to kick off our starting by pointing to the why yes, to the potential benefits for health, for economy, for environment. I want to focus on the opportunity cost of not exploring promising fields of research, production, because of the ethical principle of public interest that we sometimes forget to put at the forefront. There is stewardship of knowledge, of resources. It is in the public interest that these technologies become beneficial to everybody. And in the same context, we're often forgetting about the right to benefit from the advancement of science and its application. It's a right that is named in the Universal Declaration of Human Rights, in the International Covenant on Economic, Social, and Cultural Rights. These are documents that Canada signed and ratified. But for many years, this right was sort of dormant. It was a text that wasn't used, wasn't applied. So I think myself and other scholars believe that it's time to bring it back to life and put some meat on the skeleton of this right and start using it when we talk about emerging technologies. Another interesting principle in this context is, of course, intellectual freedom. We usually do not put restrictions on research unless we can justify them with a perceived risk that is too great to move ahead. We don't just stop research because we have a general vague sense of discomfort. At the same time, if we speak about freedom, we should remember the concept of responsibility. And then the question is, who is responsible? Do we put the onus of regulation on the government and approach things through legislation, as we did, for example, in the case of reproductive cloning? At the time that this technology became possible, most countries around the world decided to legislate in order to ban it or create moratorium. Or do we leave it at the level of self-regulation, as we did with genetic engineering in the early days, and think that the scientific community or professional societies are the best placed to regulate themselves? Regulation in any case should be always proportionate, which means it should consider risks and potential benefits, and the image is there to remind us how intricate this balance can be when sometimes the risks are unprecedented or unknown, and the potential benefits can be enormous. So I'll just conclude with a few ethical concerns that are very prevalent in the literature on synthetic biology, gene editing, etc. First of all, safety, of course. When we think about these technologies, we're always concerned about accidental or uncontrolled release into the environment, or about intentional design that is hostile to humans and the environment, and the concept of dual use. Scientists can come up with something with the best of intentions, but somebody else can use the same technology for malevolent purposes. There's the ethical concerns surrounding intellectual property. What about patenting? What about commercial monopolies? Can these create hurdles to research and also create barriers to benefiting the public? How do we handle this? Very complicated area. And a more sort of philosophical concern surrounding the concept of artificial life. Are we blurring the line between artificial and natural? Are we creating a new concept of life that is open to new interpretation? And what journalists like to talk to me about, are we playing God? A concept that I'm not particularly familiar with, is it's exceptionally vague. And to me, all of science is playing God and all of medicine is playing God. So this is what I have to say on it. What do you say to accusation that your research team are playing God? That's what's happening in your labs, I'm sure, every day. It looks like a lot of fun. My last comment is about public engagement. All the high-level policy statements about how we should govern these emerging technologies talk about the importance of open, transparent, ongoing public debate, the importance of considering the concerns of all stakeholders, including, when relevant, patients and users. And we have to keep in mind the risk of not establishing public trust and social acceptance, and the risk of backlash, as we've seen, for example, in the context of GMO. So that's another thing we need to keep in mind. And with that, I'm going back to Bettina so that we have enough time for some discussion. Thank you.
Awesome. Thank you so much, Vardit. And time flies when you're having fun. I think we had a really great sort of overview from a number of individuals in different domains. We saw also how engineering biology is really bringing this together, sort of this idea of the platform that feeds a number of different verticals. So we just have a few minutes left, unfortunately, and unfortunately the audience cannot engage with us. So perhaps we can round it up with a couple of questions that I would ask all of our panelists to give their feedback on, and perhaps the first one being, let's project ourselves in 2040, this year where the biorevolution is well on its way. 60% of the physical inputs into economy is biological. What are you excited about in 2040? Where are you going to be at with the technologies you're working on? What's the Canadian product we can buy that is based on biology? I don't know who wants to start. Peter, do you want to start? Can I pick on you?
Sure, no problem. I have to say that as my other speakers indicated, we're at a bit of a inflection point here. That what we do now will impact what we buy, or what we buy from Canadians, or what we buy from other places in the next 20 to 40 years. And I don't want to sound overly dramatic, but it takes a while to build manufacturing sectors and to build the human resources that need to be behind this. And we're in an excellent place in general. And so I really like Vardit's perspective on kind of opportunity not lost as a way to position this. To be very specific, I think we have opportunities in my area, and I won't speak for others, to really start to think of off-the-shelf therapeutics that are designed to treat disease, that are affordable, that are accessible, that can be applied in areas of Canada that currently aren't served by high technology applications. These are really important things, and we're well on our way. We have the scientific foundation for that, and I think we can have that leadership, but it won't happen by standing by and not doing anything about it.
Yeah, totally agree. Who else wants to comment?
I'll add to what Peter said. I think he's absolutely right. I like to really dream and say that we've got the ecosystem in Canada of great science, and I think the opportunity is right that we make that work for us and work for it in an effective manner to transform bioeconomy, so to say. And really for me, Canada can be a world leader in kind of public health and public good through engineering biology, because we can make affordable drugs in Canada, manufactured in Canada with innovative technologies, bedside treatments, and we've got all of the building blocks. And I think we have to just make sure we bring those building blocks together to make our beautiful Lego. I think that's kind of where we have to come together. We've got those strengths. And in terms of manufacturing, as Peter said, yes, absolutely. It takes a lot of time and investment. And there again, I think there's a great opportunity to build the HQP in this area and to invest early and really be in the forefront of bringing those type of talent to our younger generation. And the last point I'd make is that really integrating digital, because there again, I think we've got a lot of opportunity to integrate digital in many of these engineering biologic applications. And so letting that work for us through multidisciplinary approaches, I think would be a great way for the future.
Yeah, and thanks for bringing up the HQPs. We haven't talked much about it, but you know, developing that talent and those skills in this new context is really critical. I agree. Vincent, I think you had your hand up to speak.
Yeah, I think that I agree with all the other speakers, but I think it's almost like we don't have a choice. If we're going to participate in the next generation economy, we have to jump in, because the whole world is not waiting for us. So we really have to put our heads down and go. What I would say that we have to think about is what's the space that we really want to take. And as we make our decision or choices to move forward in biomanufacturing, I would say we have to look at things like sustainability and supply chains and costs as things that should guide our choices as to where we're going in biomanufacturing. And if we do that, I think we're going to make the right choices. But to me, it's not a question of if, but it's a question of when, and we have to jump in.
Yeah. The opportunity not missed by, we don't have a choice. Yeah. Adam, Vardit, who wants to go first?
I can go unless you'd like to.
Yeah, Adam, please.
Yeah, I totally agree with Dr. Martin that it's happening one way or the other. So, you know, we really, you know, we're kind of behind in Canada when it comes to biotech. I think everyone probably kind of has that same sentiment. But we're at a crossroads now with COVID kind of taking apart our economy to some degree, that we can start rebuilding it back. And the choice is, do we want to keep going the same route that we have, or are we going to actually look to the future? And so, you know, we're all here to, you know, try and instill the importance and you know, sort of the outlook for the future of engineering biology and how it's going to be applied and how it's going to be beneficial to everyone. And if we don't get in now, then we'll just be behind. And I think another example of why we really need to have our own infrastructure, have our own capacity is really, the proof is in the pudding. I mean, what happened with COVID when masks became short? Well, the United States tried to stop 3M from exporting masks, and China was, you know, holding on to them for dear life. And, you know, what would we have done if things had gotten worse, right? I mean, we can't rely on everyone else to support us, because our friends are not necessarily our friends when things get difficult, right? So, yeah, I mean, I could go on at length here, but I guess we're running out of time, so I'll go on to Dr. Ravitsky then.
Yeah.
Thank you. What I'll put on the table for my, you know, dreams for Canada in the future is not a product, but rather a governance model. I hope to see Canada choosing ways of governance that are balanced, that are nuanced. Countries in which the government decided to step into the lab found themselves with laws that were outdated very fast. Hard law is always struggling to keep up with the science. And so we need to find anticipatory approaches and regulatory approaches that are really in the best interest of Canadians. So that's my dream for Canada, is to stay away from approaches that become outdated too fast and at the end penalize the citizens.
And how do you achieve that, Vardit? How would you achieve that? I completely agree with you. I support that. So what's the path? How do you achieve that? What's the path forward?
Well, first of all, you fund researchers such as myself, because that's exactly what we study. We study anticipatory governance models that look into the future rather than base regulation on the problems of the past. And second of all, you stay nimble by not going with hard approaches. You stay away from legislation, and you try to stay at the level of regulation and self-regulation where possible, in order for your tools to remain easily adaptable. So jokes aside, there are people who work exactly in this area, because to keep Canada competitive and self-sufficient, as you guys said, we need not just the science, but the regulatory approaches that allow the science to run.
And that's so important, and also really a great justification why we need to have this group and these groups come together. So we bring those different points of view together from the get-go. So we're running out of time here. If I could just ask each one of you, if you wanted to leave the audience with one thing before we wrap up, what would that be? One word or two words that you'd like the audience to leave with?
I would say, please educate yourself on the issues.
Inclusion of all stakeholders in these debates.
Awesome. Yes.
Maybe I'll add a comment here. I have to admit, I disagree with one of my panelists. Adam said that Canada doesn't have leadership in biotechnology. You know, I think we've identified a number of sectors where we do have significant leadership. We're seeing that already through the COVID vaccine. We're seeing that in other areas. So, you know, let's build on this and move forward. I think with the NRC and with other organizations across the country, we have an awesome opportunity. So let's build on our leadership is my closing statement.
Just to clarify, I didn't say that we don't have leadership. I think I just said that biotech's a little behind in Canada, which you know, I thought that was kind of an opinion that had been sort of thrown around the panel in our previous discussions, but I just comparing it to to places like, you know, Europe, the United States, China. You know, they obviously have a bit of a head start on us in some areas anyway. But, that's all. No, I'm not suggesting we don't have leadership or a lot of potential, lots to work with. Just a comparison to some of these other countries. That's all.
Yeah. Thanks, Adam. Anyone else wants to chime in here?
I think I agree with a lot of what's said. You know, collaboration is going to be the key. I think we are good at it, but we have to be more impactful at it. So just not collaboration for collaboration's sake, but really more mission oriented and more directed in a way that we are focused on the collective impact.
Yes, I totally agree. So I'm going to thank you so much. I'm going to wrap up our session, if I can, with just a couple of final slides. Can you see the slides? So I want to thank all our panelists. I think we heard about that engineering biology is really fueling a new wave of innovations. We have seen great examples. I think I would agree with and build on Peter's comments that there is great leadership. We have all the ingredients. What we need to do is step up and act. And here again is the call to action. So on the point of please educate yourself, do read our engineering biology white paper. I think we laid the action out that are required so that we can tackle all the things that the panelists talked about in terms of, you know, funding, coming together, building on the leadership, creating the right regulatory environments, you know, training our talents and developing skills, and do all of that in collaboration. And so would ask all of our audience, please do get involved, read the white paper. You will find it on the Ontario Genomics website and also on the Can Design websites, and do get in touch and get involved with all of us. And with that, I want to thank all of my fellow panelists here. It's been great. I learn a lot every time I hear you present, which is awesome. So let's keep the momentum up and let's keep the conversation going at the National Engineering Biology Steering Committee and beyond. So with that, thank you very much and have a great day.